Well, thank you very much for inviting me, and it's a great, really honor to receive this award, and I'm really very excited and super grateful. So I'm going to tell you my life story. It will be a different talk compared to all the talks you saw. I will show you some of my signs, but not a lot, so not to overburden you. So I came to the Western world from the country that doesn't exist anymore, from the Soviet Union. It's now Russia. I came from the city that doesn't exist anymore. It used to be Leningrad. It's now St. Petersburg. I was raised as a musician. Actually, I went to specialized music school. I play piano since I was 5 years old and was supposed to be a pianist. In fact, I even play with orchestra. I was not bad, and I was sure that that will be my life. And one day, one of my teachers, she is a conductor, used to be a conductor teacher in my lab, in my school, came to me and said, "So what are you planning to do with yourself?" Well, I said, "What do you mean? I will be a concert pianist." And she said, "Do you think you have what it takes to be a world-known concert pianist?" And I have to say that I was kind of shocked because nobody ever questioned it. Everybody just thought, "Well, you're just so great, you're so good." So I was sure that I can do it. But then she said, "You know that it's not enough to be good or great even. You have to be one in millions in order to survive for a long time as a concert pianist." And I have to tell you that I didn't sleep that night. I was crying. I was sure that my life is over because I understood that I was not that great. And I realized that I could not make it for the rest of my life to be at the top of that profession. So, but in the morning, I made the decision. I decided, I was already 3 months before I would graduate from this music school. I decided to drop that music school, went to the evening school to get my high school diploma, worked as a pianist teacher to earn money, and hired tutors in chemistry, in physics, in math, in order to enter the university. I decided that I don't want to be an engineer. It was just not in me. I didn't want to be a physician because my parents were physicians. I kind of decided, okay, everybody likes nature. I will find something. So that's how I became a biologist. So I was lucky enough, I was able to pass exam and enter the university in biology department. And then I was surrounded by all the students who came from specialized math school, from specialized physics schools, and I came from specialized music school. So my life, especially first 2 years, were very difficult because I had to work super hard to get into depths of knowledge compared to all of them. But anyway, I managed, and I was able to, so I specialized and worked my master's degree and then PhD were on unicellular organism called ciliates. And these ciliates are very, so I work on ciliates that actually live in different seas. So I travel quite a lot. And when I defended my PhD, I got a job at, as an assistant professor at the Institute of Academy of Science in Leningrad, Institute of Cytology. So, but as I said, I traveled a lot, and at that time we had no money, and at that time we already had our little Eugene, who is already not so little anymore. He is sitting right here. So we traveled a lot with my husband. I would go into expedition, and he would come, and we would spend a week traveling after my expedition was over. And once It was in 1978. I went to expedition in Caspian Sea in Baku. And after that expedition, my expedition, Alex came with Eugene, and we traveled for a week. And on May 18th, we were flying back home from Baku. But what happened? Our airplane crashed between Moscow and Leningrad on a potato field. And many people were killed, but we were very lucky and we survived. And so then, this time, and actually I have to tell you a little bit more. So I didn't like Soviet Union. I didn't like the country. I always wanted to immigrate and live. But it's difficult to leave the country where you have all your relatives and everybody is, who you know. But this crash was kind of last drop. Well, we decided that, well, we could have been killed. We have another chance. We are going to immigrate. And both my husband and I were Jewish, so at that time it was possible to immigrate from Soviet Union. And so at that time, in order to immigrate, it was customary. People who would like to immigrate would come and ask to be fired, because otherwise your professor and all your colleagues will have problems. So I came to my professor and I said, listen, such and such, I decided to immigrate, so I would like to be fired, knowing for sure that in Academy of Science, if I would not get a permission to immigrate, I would have never, ever got this job back, period. He looked at me, he said, are you crazy? You want to leave? You are in Academy of Science, you live in downtown Leningrad, you want to immigrate? Where do you want to go? Well, I said, I want to go to America. And he said, you will never, ever make it in America. You will be sweeping streets of New York, and Eugene will be selling newspaper next to you. And so I said, well, if that's the case, probably that is going to happen, but I want to try because I have another chance. So he fired me, and the same happened to my husband. And we were actually lucky because at that time, Carter was president, and he was pushing Soviets to let more and more Jews to leave the country. And, and then Russians were giving grain for free because it was starvation at that time. So we were lucky because in 4 months, they told us in April 26 that we have to leave the country in 3 days, period. So we left the country and came to Vienna. That's how was immigration through Vienna, and then to country where you want to go to. And we wanted to go to the United States. However, we didn't know how to apply for a job. In 1979, it was just a very beginning of immigration from Russia. And I knew only one American scientist who came to my institute. Actually, he came the day we dropped from the sky. He arrived to the institute, and his desk was next to my desk. So I kind of knew him and his wife because they spent sabbatical in Leningrad. And I asked, so when we came to Vienna, I wrote to him and asked, how do people apply for a job? How people, what to do? And by the time we arrived to Rome, we received a telegram from Jerry Polin, who was a professor at the University of Georgia, Athens, Georgia, America. And in the telegram, he said that Alex, my husband, has an offer as a postdoctoral fellow in entomology department, and I could have a part-time postdoctoral position in his lab, working with protozoa. That's the reason, and that's how we came to Georgia from downtown St. Petersburg to Athens, Georgia, 40 years ago. It was very, very hot. We never ever experienced such a hot, heat and humidity. And my poor mom, who immigrated together with us, asked me outside, "Are you sure that you didn't mix up countries?" And I said, "Well, you know, Mom, please." And so we started new life, basically, again, as a postdoctoral fellow. We arrived to Athens with $25, all between 4 of us. So so I started to work in Jerry's lab, and during the first year, I even published a paper, but was not interesting because I published a paper on similar things I've done before in Russia, only different species. And it was just kind of blah in zoology department. However, I noticed that botany department in Georgia was very vigorous, very interesting. People had a wonderful seminars. They were talking about molecular biology, genetics. And as soon as I got an offer to be a postdoctoral in one of the lab in botany department, I immediately accepted and moved to botany department. And that's how I became plant biologist. So from musician to working with ciliate and then plant biology. Again, from starting my lab, I always tell my graduate students and postdoc, change is good. Don't be afraid of change. Anyway, so we spent, so when we were in Georgia, so cell biology in Georgia. So in Georgia, in botany department, I learned how to clone the genes, how to make monoclonal antibodies, polyclonal antibodies. So I've learned a lot of things I had never had any idea back in Russia. So it was very stimulating, very interesting. Also, my first gene that I cloned called with gamma-glutamine selectin. Also in collaboration with graduate student Michael Mishkin, we were able to find that these lectins are expressed very unusually. At least at that time, it was a very good question. They expressed, it depends on, first of all, they expressed only in monocot, grasses, right? Like in barley, it will be only, you see, one cell layers of a tissue called coleoptile. It doesn't matter. You see, one cell layers. Then, for example, in rye, it will be 2 cell layers. And for example, in rice, it will not be expressed altogether. Basically, this observation at that time was very kind of important because it was cell type specific expression of similar genes in different species. And basically, this observation gave me a real job in America. I will tell you in a second. So we spent 7 years in Georgia because by that time we already had another son. He's just arrived and he's sitting over there, my baby. And so by that time we had to, and we didn't want to live in different part of the country. And so we stayed in Georgia for 7 years until, and we would get an offer, but it will be one part of the country, the other, so we stayed there. One day I applied for a job. I found a job advertisement in science at Plant Research Lab in Michigan State University. And Michigan State, this Plant Research Lab, people who know plant biology knows probably that it was a really very premier institute in plant biology in terms of reputation and science. By that time, but I didn't really know how great it is. But of course I applied because, you know, and they asked me for interview. I came and I said, I gave a seminar and then I said, I will be studying molecular mechanism of this cell type specific expression of these genes. But I have to tell you that in these years, in '83, we barely knew how to transform tobacco. Tobacco doesn't have these cell layers, and tobacco doesn't have this gene. Monocots, even now, with all our ability to transform things, we can't answer this question. But the question was a good one. So I was pushing an envelope. And the place in Plant Research Lab was incredible. When I came and I gave a talk, I saw it was completely international. And after Georgia, many of you don't know what, it's very homogeneous. It's extremely, so as soon as you stand out a little bit differently, especially 40 years ago. So I loved the place. I wanted that job so badly. And suddenly, so later on, when I became mover and shaker in that plant research lab, I understood how I got the job. The people who were in charge there, I think they just decided, look, she looks different. It looks like she's trying to do something she cannot do, but the question is so good. What if we give her a job? And if she doesn't make it, we will not give her tenure. And basically, I'm sure that's why I got a job. Because I knew I didn't come from anything super duper. I didn't come from famous place. I mean, and I was proposing something that I really cannot do. But they give me this opportunity. And so, of course, when Hans Kinde called me and said, oh, we're going to offer you a job, I said, oh, I love it so much, but you know, I have a husband. So anyway, it was typical. So husbands sent his documents to Michigan State. To make a long story short, they liked him so much, they gave him associate professor position. And I got assistant professor position, but it made no difference to me. I was very happy. And that's why, and then we moved that's how we moved to Michigan State. And we put our both children into our Chrysler car and a cat, and we had a little trailer, which is the main possession of a trailer was huge jar with liquid nitrogen full of monoclonal antibodies. These days, nobody would ever allow us to do anything like that. And we moved from south of the country back to the Midwest, and that's how we started our new life. As an assistant professor, I was 39 years old. Again, just to give you, especially young people, perspective, don't despair. You don't have to start everything at 30 or 29. You can make it even when it's later in life. Anyway, so I started to work in this plant research lab, and at the very beginning, I had a wonderful two graduate students, Thea Wilkins and Sebastian Bednarik, and what three of us made an observation that this lectin with gemmaglutamine, it's a very simple slide. I just will tell you, for those who are not biologists, simply, you see, this is, when you analyze it on a gel, you can see that it's migrate shorter than you would expect from its sequence. And to make a long story short, what we were able to find that this what called C-terminal propeptide is cleaved, and it's a mechanism like keys and lock and key to send it to the vacuole. So basically, to understand how to go to traffic within a cell. And that made a difference in my life. My life became a trafficking lab. I didn't study cell type specific expression. Again, be attentive to nature, pay attention, and just have intuition what kind of question to ask. I didn't study what I was asked to, said that I got a job for. That's how I'm known, basically, as a trafficking woman, trafficking scientist, because that's what I study. This was, it was not known before, and how proteins get to the right destination. And that's what we study. So if you take out this, what is in green, the protein will be secreted. So it's basically very important address. And later in my lab, we had many postdocs and graduate students work on different steps of machinery. Here you can see a very schematic representation of what's going on in a plant. It's endoplasmic reticulum called organelles, Golgi, vesicles, vacuoles. And in order to get from one part to another, you need always, it's a key, you need to know the key and you need to get into the right locker. So, and it's called machinery. And we have identified many components of this machinery. So, by that time, and again, I'm doing very briefly all this scientific work and everything because just I have only 25 minutes. By that time, I was approached, first of all, I have to say, not me. My husband was approached by University of California, and he really wanted to move from Michigan to California. And I didn't want to move, actually, because I liked Plant Research Lab. It was so nice and wonderful. But again, I have to tell you, I pushed myself, and it was the best decision I have made, because I have developed very differently in addition to my science. And I will share it with you in a moment. So we both moved to University of California in Riverside, and the way it worked for me, the chancellor of that university asked me, said that, you know what, I want you to help me to put this university on a map, at least in some area. What can you do? Can you just do the cell biology? So I will give you resources, and that's how I started to develop as a leader there, okay? At first, I will tell you a little bit about science we've done there, because it also was very different. I started another approach called chemical genomics. And the reason why we did this chemical genomics is because many genes in this membrane trafficking are what's called redundant. For every function, 5, 6 genes. So genetic approach is very difficult, laborious. So we decided to try chemical genomics, to use a small chemical compounds to perturb function. And we have identified many chemicals that are important for trafficking in particular parts of the cells. And some of them we called endocytins. And I will tell you what we found only about one chemical called endocytin 2. It stops trafficking of vesicles from this part to the plasma membrane. And it doesn't allow the content to be secreted. Why it's important? So, because this endocytin 2 binds the important protein called EXA70. So, for those of you who are not plant biologists, just to tell you, it's not so simple in plants. You have a vesicles, and then you see how many proteins. The whole component has to move these vesicles to a right part of the membrane. And that's how content of these vesicles could be delivered to outside. So this EXA70 is a key component of this so-called EXA-cyst complexes. An EXA-cyst complex was implicated in several cancers in mammalian cells, in human cancer, sorry. So ES2 is effective in mammalian cells. It can bind to EXA70 homologs in vivo. So now many pharmaceutical companies and many medical scientists using this our chemical, trying to modify it and make a better connector, better binder, and use it as a drug, because not a single drug so far available that binds specifically to EXA70. So this is just one vignette telling that it's how important it could be. But as you remember, the chancellor hired me to build something special in UCR. And what I decided to build is to build a center for plant cell biology, but to build it in such a way that it will be first not only for plant people. So it was umbrella for plant biologists, but all biologists and even chemists and physicists, they were coming and using our facility. So I had two approaches. We built a fantastic core facility, microscopy, proteomics, genomics, bioinformatics, metabolomics, and we had, and my other main objective was to hire the best PIs. So, and I have to tell you, I can't tell you how grateful I am to this. 10 years later, we celebrated our anniversary, and by that time, this center became known one of the best center in plant biology in the world. So it's very rewarding, and I'm extremely proud of it. Also, what happened to me back in Michigan when I was in Plant Research Lab, I was asked to become an editor-in-chief of a journal called Plant Physiology. It's a journal of very wide, cover very many areas of biology. And when I became the editor-in-chief of that journal, the impact factor was 2. I put a lot of effort, and I very much excited about it, and I loved that work. But in 5 years, we increased the impact factor to 6. For a journal of wide scope, it's a big thing in 5 years. But after 5 years, I decided that I already was in UCRI, I was very busy there, and I wanted to stop there, and I decided, didn't want to do it anymore, although they asked me. And I was concentrated back on CEP Institute there. So another thing which was very interesting, again in this Center for Plant Cell Biology, when I came to University of California, we had only one man who was a member of National Academy, and he left. Then right now we have 5 UCRI members who are members of National Academy, and 4 of us are female. You see, this is Sue Vessler, me, Julia Belliceris, and Shamei Chen. And last year, Sean Cutler joined us, so it was elected last year. And to the best of my knowledge, in the United States, it's the only place that has 4 females in the same place as members of National Academy. So I really think it's wonderful. So by that time, so when I turned 70 2 years ago, I decided that I want to retire. I didn't have to retire. They want me to stay, but I wanted to retire because I wanted another change. I wanted to do something else. So what do I do now? So I am on quite a few science advisory boards, a lot in China, 3 or 4 in Europe, and some in the United States. And I am associate editor of PNAS at National Academy of Science Journal and editor of several other journals. Here's just a picture of where I'm head of the board of directors of the institute. It's a new opportunity. We built an institute between John Innes Institute in England and 2 universities, from Peking and from Shanghai, make international institute that located both in Shanghai and in Peking. So it's an example of a new type of science in China, and I've been there from the very beginning. We started it 5 years ago. My other thing is, what I do is continue promotion and support of young scientists and women in particular. And for this, I have to tell you one small example how we operate in the United States. In 1992, it was a Gordon Conference. You probably have heard about Gordon conferences, right? They are, very prestigious. And in 1992, this woman in the middle, Vicki Chandler, was the only female who was giving a talk, was invited. So, however, she was elected to be a chair of the next 1994 Gordon Conference. And she invited 7 of us to be chairs of the symposium. And so we selected other people to give talks. And during this meeting, one of the guys took our pictures, and then we hear they said, wait a minute, this is a power woman. What did we create it? What happens? But I have to tell you, we are not power women, but we have enormous influence, and we are a very tight, friendly group. We support one another, and we support and help young people all over who come across, who we can notice, not only women, young people, but we watch after women very, very especially. And we are extremely friendly. And I have to tell you, out of 7 of us, 6, it's the 7 of us, right? 6 are members of the National Academy. So you remember that I started with music, right? And music helped me so much. And I love music and I love art. And it helps me a lot in life. And I want to share it with young people. So I came with the idea to create I call creative disruptions. During scientific meeting, we would organize small group of local artists will show their art, and like 15, 20 minutes music will be performed. For example, this is what happened in China when I'm head of the International Advisory Committee. So they would play Chinese classical music and Western music, and everybody loved it. And I think it's enriching, it's educational, and it's extremely important. So, as I said that, I decided to retire, and my people organized a symposium for my retirement. Some of them were able to come, and this is the picture of my scientific family, my former students and postdocs. They all prepared me, gave me a nice present. It was framed piece of art with people they were able to find who went through my lab. It was just wonderful. I love it. It's in my bedroom. And this is my biological family. This is my treasure, two of my sons. This is Eugene. He's sitting here. He's associate professor in the University of Chicago and anthropologist. And he and his family live in Chicago. And my little one, Vincent, he is a physician at the University of Washington Medical Center. And they both, he and his wife and 2 children live in Seattle. And they are just unbelievable. They are they're my life. And I will finish with last slide, which means a lot to me. So in Europe, we have a regular meeting called NPER meeting. It's a plant cell biology, European plant cell biology meeting. And I attended that meeting from the very inception, I think, 20, 25 years, right? And it was, I think, meeting in Bordeaux. Right after my retirement. And during the banquet, suddenly they presented me with this painting. And the artist was shown my picture. You can recognize me a little bit. And I'm sitting on a textbook in Russian and English textbooks. I'm playing piano, but chemicals comes out of my playing. My Golgi and nucleus, all organelles are within the piano. And of course, women represent my dedication of helping and supporting women. And a lot of thought went into this painting because in Luxembourg, I bought this piece of art, and it's now a leg of my piano. I adore it. It's in my bedroom in front of me. Whenever I wake up, the first thing I see is this picture. I am so grateful to my European colleagues, and it's just incredible pleasure to have it. And that's it. I will be happy to answer your question if you have a question.